The question is, give differences between the following. The first difference is between Chlorenchyma and aerenchyma. Both are a specialized form of parenchyma. Chlorenchyma is parenchyma tissue having chloroplast, so it can perform photosynthesis. And aerenchyma is a parenchyma where you can see large intercellular spaces or the air spaces chambers present in between the cells. Chlorenchyma primary function is photosynthesis as it has chloroplast which helps in converting light energy into chemical energy, that is the sugar glucose. Aerenchyma, since it can trap more air molecules, it facilitates in gaseous exchange, that is oxygen and CO2, between plants' shoots and root, especially in waterlogged conditions. It also helps in providing buoyancy in case of aquatic plants so that it can float. Second difference is between tracheid and vessels. Both are the elements of xylem tissue. Tracheid is a single cell, whereas vessel is made up of a group of cells which are arranged one above the other. Tracheid's end walls remain intact, whereas in vessels the end walls get dissolved, that is where the two vessels get connected. So if I take this is a vessel element and that is the another vessel element, the cell wall that is present in between two vessel elements will get dissolved and becomes perforated, so water can easily move in these holes. Then we have tracheids. The walls of tracheids are very thick with a narrow lumen, whereas in vessels they are less thick and the lumen is wider. Third difference is between blood and lymph. Both are fluid connective tissue in our body. Blood is red in color, whereas lymph is white or colorless. Blood is red because of presence of RBCs, that is the red blood cells, whereas lymph do not have these RBC cells. Blood has high concentration of protein compared to the lymph, which has a less protein content. Blood contains fibrinogen, which is the clotting factor. Lymph contains less of these fibrinogens. Blood contains more quantity of digested food and waste because it helps in transporting them, whereas lymph contains less of this digested food. The main digested food transported by lymph will be the fat molecules or the water insoluble molecules. Next difference is between bone and cartilage. Both are proper connective tissues. Both are helpful in skeletal structure. Cartilage is firm but little elastic, whereas bone is brittle. The matrix is formed of protein known as chondrin in cartilage, whereas it is made up of more salts, or one protein is osin in bones. Inorganic salts formed of calcium phosphate crystals are deposited in the bone. Cartilage does not have a lamella, whereas bone has a structure known as lamella. The cells in the cartilage, that is the chondrocytes, occur in group in of 2 or 4 in a space known as lacuna, whereas in bone the osteocytes are present in form of a concentric ring and they occur singly inside a lacuna and they are arranged in concentric circles in the lamella. Cartilage, there is less supply of blood that we can see, and in bone we can see more supply of this blood vessels. Next difference is between tendon and ligament. Both are again connective tissue needed at the joints. Tendons formed of strong and non-elastic tissue, whereas ligament is formed of strong but elastic tissue. Tendons joins voluntary muscles to the ends of the long bones, so it connects bone and muscle, whereas ligament connects to bones. Tendons have white fibers formed of collagen protein, whereas ligament has yellow elastin fibers made up of a material known as elastin. Tendons, the fibers are long and occur in parallel bundles, whereas in ligament, fibers are small and are found scattered in the matrix. Next difference is between the meristematic and permanent tissue. Meristematic cells, we know they are small and they are undifferentiated and their main aim in the life is to divide. So they are living throughout their life and they are dividing throughout their life. Whereas permanent tissues, if we see, cells are large and differentiated to form a particular function, and they may be living or non-living, but they do not have the power to divide. Meristematic tissue, they are very compact, small structure, so you will not see any intercellular space. In permanent tissue, there will be often intercellular space seen. Meristematic tissues may or may not have vacuoles, whereas permanent tissue has large central vacuole. If we see the cell wall in meristematic tissue, it is thin and made up of a cellulose material only, primary cell wall. Permanent tissue, cell wall may be thick or thin depending on which type of permanent tissue we are talking about. It can have depositions of suberin, lignin, pectin, so on. In nucleus, if we see, meristematic tissue has a large central nucleus, whereas in permanent tissue, it will be pushed towards the side because of the large central vacuole that is present. So, nucleus is relatively small and lies to the periphery region, that is the side. I hope it is clear to you. Thank you.